Hey y'all, how are you all doing today? So last time, what we're going over is what we went over was simply Mendel's hypothesis and how it went against the normal hypothesis for genetics for hereditary traits, which was the blending theory of heredity. And basically, what he did was he looked at the final. He took a true breeding flower, which was blue. So this was a true breeding blue flower in the parental generation, and he crossed it with a true breeding white flower. And basically, what he got in the F1 generation was all the flowers were blue, which was not expected out of the blending theory of heredity, right? And I'm going to leave a note like on the top right of your screen to look at that video if you want to. And then for the F2, he crossed the F1s together. And then in the F2, he ended up with a ratio of 3 out of 4 of the flowers were blue, and then 1 out of 4 of the flowers were white. So basically, in this section of the chapter, we're going over is how did he get these ratios, this 3-to-1 ratio, and what hypotheses has he made, and how did he validate his theory. So what he came up with is something called a particle of inheritance. This is also known as an allele. And what he hypothesized was that every individual had 2 alleles, and each individual can give either allele to their offspring. So this is basically what he was hypothesizing, was that each of these flowers, they had 2 alleles, and then each of these flowers can give either like the dominant allele or the recessive allele. And that was basically like his assumption. And then this is also referred to as the law of segregation. And it's basically during gamete formation, alleles from a gene will equally separate from one another such that the final probability of each allele is half and half. So basically what this means is that if you have an individual like big A, little a, to get big A, probability is simply just 1/2, and to get little a is also 1/2. Same thing for big A, for a homozygous individual. Because you have 2 big A's here, they would be half big A, half big A. So basically 100% big A. And then same thing for little a, where you're going to have 100% little a. Okay? So how did he validate his hypothesis? what he used is something called a test cross. So a test cross is used to determine the unknown genotype. Is used to determine the unknown genotype of an individual by crossing them with an individual that it is recessive for the trait, homozygous recessive. And what you can do with this is you can use it to figure out if your unknown individual has, the big A genotype or big A little a genotype. You're not going to really use this for an individual that is recessive, because the only possible genotype that they could possibly have is just little a. So basically what he's doing here is he hypothesized that you would have two alleles, and what you could do is you would get a data set which we're going to go over in a couple of minutes as practice. And then from that data set you can determine that, that this in fact is two different alleles that's causing this trait. And then what he did next was he looked at he predicted that the flowers that were blue, he predicted that 2/3 of them are actually heterozygous, and they included both of the alleles, and then you had 1/3 of those that were blue are homozygous. So what he did was he essentially ended up crossing the F2 together again, and he found plants that were homozygous for the blue flower, homozygous dominant for the blue color, and he crossed them together, and he ended up with a ratio that was basically 100% blue. So that doesn't really tell you anything. It just tells you that if you cross these together, you would have blue flowers. So there will all be big A. But then what he did also was he took the blue flowers that were heterozygous for the gene, and then he crossed them together. And what he found was that you would end up with the ratio that we saw before there, which was 3/4 big A, whatever, and then 1/4 little a. So basically this is like his assumption that, if you had different genotypes right here, you're going to have different ratios that you're going to get from the following cross, and then that will confirm that, this is in fact two alleles that's causing this this inheritance pattern. So basically this is essentially what he did, and the way how geneticists, the way how you're introduced to do these crosses is simply you would have your genotype. Let's say we're crossing big A little a with big A little a. So the way how this will happen is you would use something called a Punnett square, where you're gonna write it in this sort of fashion. And I should change this color. So we're gonna change this to blue so this would be parent number 1. This would be the female, and then this would be the male here. So what can happen is the female can either give big A and the male can give big A, or what can happen is the female giving big A but the male giving little a in this situation. Same thing for the male, he can give big A and the female give little a, or both of them can give little a, little a. So basically, how do we actually know that the proportions of these individuals from the following cross? And usually what I get from students is that they say, you know, to get big A, it's 1/4. So big A. To get big A, little a, it's 2/4. And then 1/4 little a. And whenever I ask them, like, why is it that you are getting, you know, 1/4 big A, they usually just explain it as, you know, there's 4 squares here, and you only have one possibility. Out of those 4 squares, you only have 1 square that is big A. So, therefore, big A has to be 1/4. And that is the exact opposite way how you should be thinking about genetics from now on, okay? What you want to do is, remember the law of segregation that we just talked about a couple minutes ago? From an individual, from the male individual in the red, that male can either give big A or little a, right? The probability of that male making a big A gamete, it's 1/2. And then same thing for little a. So with that same assumption, that female can also make 1/2 big A and 1/2 little a. So what you can do now is you can combine these together. So if we're trying to combine big A and big A from both the male and the female, we would multiply that 1/2 by 1/2 to get big A is 1/4. Same thing we're going to do with the big A little a, 1/2 times 1/2, big A little a, 1/2 times 1/2, it's big A little a, and then 1/2 times 1/2, it's little a. And then in genetics, whenever you have a situation where both of these genotypes are identical, that's a sign that you should add these fractions together. So 1/4 plus 1/4 is equal to 1/2, or also 2/4 as well. So that's essentially how you get these sort of crosses. And I recommend students to just memorize all the different possibilities that you can get for a single gene cross. You can have some other instances too. We can try some different ones as well if you want to. So let's try a different one. We can try big A with big A little a. So to get big A from this individual is 1/2. To get little a is also 1/2. To get big A from this parent, it's all just big A, right? So what you can do is to get big A from this cross and to get big A little a, what you're going to end up with is, so 1/2 times 1 would be, it'll be 1/2 big A, and it'll be 1/2 big A little a. So in this situation, they have a different genotype. So the genotype ratio, or the genotype frequency or ratio, we can say as 1:1. But the phenotypically these individuals are the same. So the phenotype will just all be big A, whatever that in that situation could be. So basically, this is the way how you can perform the crosses. So let's go over some more practice problems. Typically, students don't really struggle with this section, but But remembering the basics is going to be key, especially when you're not doing just a single gene cross, but when you're going over multiple gene crosses with other types of factors as well, that we're going to build upon what we're learning about in this video. So let's say you're an actual geneticist and you're out in the field trying to get some data. And what you're going to do is you're taking, you know that there's a single gene occurring over here, and you're taking a true breeding circular tomato and you're crossing it with a rectangular tomato that is true breeding. So our parent generation is circle crossed with square. And then in the problem it tells you that all the F1 are rectangular. And then you cross the F1 together and you get the following data. So it should be around 61. So you get the following data and you're trying to figure out what the mode of inheritance for this trait is. So this is a single gene. This is a single gene because the problem tells us. But we need to figure out exactly how we're getting this ratio and what kind of thing can we come up with that. Or not ratio, we're getting a complete data set basically. So from this data set, what people typically start off with is they do 45 divided by the total, and they get some sort of a number, and in this case should be around 7.3, something like that, and then 16 divided by 61. They get some data that is like around around 0.27 or something like that. And basically they look at that data and they're like, oh, so it's basically three-fourths of them are rectangular and then 1/4 of them are circular. And that's fine. You can do this problem this way. But the issue comes up with the reason why I recommend don't try to find the frequency number, but rather find the ratio number. The reason why I mention this is because when you look at a frequency number such as something around like 0.5, 0.5 may not necessarily mean that your number is that you're getting a ratio of like 1:1 or something like that. Basically, a 0.5 could mean that you're getting something like that you're getting either it could be 1/2, so a 1 ratio, or it could also mean that you're getting something as a ratio of 2. So in other words, the way how you can think about this is if you try to find the frequency of something, you're actually going to get a false hearing where you might think that something's actually just, 1/2, like a 1:1 ratio. Like if you get something like this means that you're getting a 1:1 ratio, whereas this may mean that you're getting a 1:2:1 ratio. Obviously in this problem, like you're going to see 3 different possibilities, but the more problems you do, the more chance you have to realize that finding the frequency will not get you anywhere, and you should always find the ratio. So the way how you can find the ratio from a data set like this is that you would take the frequency number that you got, so 0.73, and what you would do is you would multiply it by the denominator of a single gene cross, of a monohybrid cross. So basically, in other words, is a monohybrid cross is, you would have a data set like 1/4 will be big A, and all this means is the 1/4 is that you would have one individual, so this will be a person, out of 4 total will be big A. So what you can do, and a frequency is basically the same thing, where you have, this is individual, and that'll be the total. So what you can do is you can multiply this by the denominator, so we can cancel out the total. And you would end up with a ratio of around 3. So this is a way how you can calculate ratios from these following crosses. From a single gene cross, because we're doing a big A little a with a big A little a cross, we're going to use the denominator here to multiply by the frequency to figure out what the ratio is. And same thing with the bottom one too. The bottom frequency for the circular one, which was 4 again. And if you do this calculation, you're also going to end up with 1. So you end up with a 3-to-1 ratio. And so basically what this means is that the rectangular phenotype is dominant over the circular phenotype. So in other words, you need 2 circular alleles to be circular. And so far we can't really do much with this problem. This was sort of an introduction to how you should think about these problems with try to find the ratio. And always look for the ratio for these problems. That will give you a better foundation than if you were to just not look for a ratio. Okay, and then the last one is you're performing a couple of test crosses. And remember what a test cross was? A test cross was you're crossing an unknown individual with an individual that is considered recessive, homozygous recessive, and that'll give you some data sets. So you have one unknown over here where you're getting approximately equal numbers, so 43 to 45. So how do we know what genotype our individual is? And remember what I said, that if you're trying to do little a and you're crossing it with a test cross, you're not really trying to figure, you're just wasting your time, because little a, the only possibility for that phenotype is just little a. So what we're trying to do is we have 2 individuals that could be either big A, or big A, and we're trying to figure out which one is which. So what we can do is we can cross these together. So we have big A crossed with little a. So you would have a half chance to get big A and a half chance to get little a. And we're going to cross this with a 100% chance of getting little a. So then what you would have is you would have a 1/2 chance to get big A, and a 1/2 chance to get little a. So basically a 1-to-1 ratio. So our unknown one was basically a 1-to-1 ratio. So our unknown one had to be big A, little a. Let's change this to, I made a mistake right there. We're going to change that to 80 individuals. So for the other one, we're trying to figure out if this is big A, big A. So we will cross big A with little a, little a. To get big A, you would have a 100% chance. And to get big A, little a, you would have a 100% chance. So all of your individuals from this cross should be big A, little a, or 100% the same phenotype. So that'll be how you would do test crosses. And that's basically it. That's basically it for this section. Typically, people don't really struggle with this, so my recommendation is sort of understand like the simple basics of the crosses and how you do them, and then as we go into multiple gene crosses, that's where like stuff gets tough. So hopefully you can refer to this video when you need some help.